Hi everyone, if you may see this, Dr. Phil here. I'm just going to go over the McFarland latex tube before tomorrow for lab or for anybody in the subsequent classes. So I just wanted to let you know this is the standard for turbidity of the culture. So if you remember from the beginning of the course, we were talking about the different types of media. That is a nutrient broth, and that is relatively clear, so we know that there's nothing growing in there. There's another one that's clear. But if this theoretically was something that we inoculated, because of the turgor or the color, we know that something is growing in there. And once again, I said I don't know if it's everything's viable in there that's alive, because we can't take the bacteria's respiration or pulse. We don't know if they're all alive at that point. But what we're going to do is we're going to take our plate, and I did some staph epididymis, gram-positive, and I'm going to inoculate one of these tubes so I can get the same, more or less, same turbidity of this, so I know we have a standard. So when we go to inoculate, excuse me one second. Tomorrow when we go to inoculate our plates to check our antibiotics or our disinfectants, we will all have the same concentration on the plates. So we have a standard. So what I'm going to do is I'm going to flame the loop here, and this should get red hot theoretically. So I can say that's probably pretty sterile. Right. I'm going to flame my tube. And this is pretty cool. I'm going to just grab some inoculum off of that plate, and in there, and that probably is not enough. That should be sterile, but just in case, we'll just hit it again. I'll cool it, grab some more. Could be close. I'll check it again, but that seems to be probably like there's too much in there, so I'll have to dilute that. But what we want to do is we want to make sure that this tube and this tube have the same turbidity. So then tomorrow we're going to take these antibiotic discs, and we're going to lawn the plate with the bacteria. We're going to test it against Staph epi gram-positive and then E. coli, which is gram-negative. We're going to take our discs and we're going to place them on the plate once it's been inoculated, and we're going to look for those zones of inhibition. So what I did is I made a bunch of these sheets up for everyone. So we're going to grab a bunch of different antibiotics, and we're going to use different modes of action for these. So whether it is a protein synthesis inhibitor that inhibits metabolic pathways, cell wall inhibitors, like the penicillins, vancomycin, bacitracin. We'll do cell membrane disruptor, so the polymyxins, or we can do the most effective generally are the DNA and RNA synthesis on the ribosomes. But I've made a list for everyone. I'll post this to Brightspace, but I just broke down a bunch of the different antibiotics. So you can just pick 3 or 4 of these if you want, so we can see how the different antimicrobials work and their modes of action. All right, so hopefully, yeah, I would say that's definitely too turbid. So what I'm going to probably do is pour this into there, and we will just this one get rid of it anyway. So. in a perfect world, these two would have the same turbidity. They're close, but I would say it's probably a little bit on the denser side. But now I have a consistent concentration in there, and we all draw from that. It's not going to really matter.